Hello and welcome to homeostasis. Okay, so to function efficiently, organisms have control systems to keep internal conditions near constant, and this is a feature known as homeostasis. So of course this requires information about conditions inside the body and the surroundings, which are detected by sensory cells. And there are various sensory cells depending on which component of homeostasis we're considering. So some of the physiological factors controlled in homeostasis in mammals are core body temperature, metabolic wastes, particularly carbon dioxide and urea, blood pH, blood glucose concentration, water potential of the blood, the concentrations in the blood for the respiratory gases, oxygen and carbon dioxide. So first we're going to look at the need for animals to maintain a stable internal environment, and then we'll consider how they maintain a constant body core temperature. Okay, so the internal environment of an organism refers to all the conditions inside the body required for cell function. For a cell, its immediate environment is the tissue fluid that surrounds it, right? Three features of tissue fluid that influence cell activities would be temperature, right? So low temperatures slow down metabolic reactions. At high temperatures, proteins like enzymes are denatured and can't function. Also, water potential. So if the water potential decreases, water may move out of the cells by osmosis, causing metabolic reactions to slow down or stop. If the water potential increases, though, water may enter the cell, causing it to swell and maybe burst. Okay, so the third would be concentration of glucose. Glucose is the fuel for respiration. So a lack of it will cause respiration to either slow or stop, depriving the cell of an energy source. Too much glucose, though, may cause water to move out of the cell by osmosis, again, disturbing the metabolic process of the cell. So in general, homeostatic mechanisms work by controlling the composition of blood, which therefore controls the composition of tissue fluid. Most control mechanisms in living organisms use a negative feedback control loop to maintain homeostatic balance. This involves a receptor, okay, or a sensor, and an effector, which would include muscles and glands. So the receptor detects the stimuli that are involved with the condition being regulated. A stimulus is any change in a factor, such as change in blood temperature or water content of the blood. The body has receptors which detect the external stimuli and other receptors that detect internal stimuli. So these receptors would send information about the changes that they are detecting through the nervous system to a central control in the brain or the spinal cord. And we pretty much know this. We learned about this in STEM. But this sensory information is called the input. Okay, the central control will instruct the effector to carry out an action, and that action is called the output. So these actions are sometimes called corrective actions, as their effort is to correct or reverse whatever change occurred. Continuous monitoring of the factor by receptors produces a steady stream of information, which goes to the control center that makes continuous minor adjustments to the output. And as a result, the factor, whatever it is, like blood temperature or water content, will fluctuate around this particular ideal value. This mechanism to keep changes in the factor within that narrow limit is called the negative feedback. So in these systems, an increase in the factor will result in something happening that makes the factor decrease, right? And similarly, if there is a decrease in the factor, then something happens to make it increase. Homeostatic mechanisms involve negative feedback as it minimizes the difference between the actual value of the factor and the ideal value or set point. Okay, so again, homeostatic mechanisms involve negative feedback because it will minimize the difference between what is occurring that's throwing your factor off and what it actually should be. So again, these factors, they don't stay constant. They will fluctuate a little above and a little below. And the homeostatic mechanisms in mammals require information to be transferred between different parts of the body. And there are two coordination systems in mammals that do this. It's the nervous system and the endocrine system. So in the nervous system, information in the form of electrical impulses is transmitted along nerves, right, along neurons. And the endocrine system uses chemical messengers called hormones that travel in the blood in a form of long-distance cell signaling. Okay, and again, we learned about this to a lesser extent in STEM, and we're really just getting really into detail about this. Okay, so thermoregulation is the control of body temperature, and this involves both coordination systems, nervous and endocrine. All mammals generate heat and have ways to retain it within their bodies, and they also have physiological methods to balance heat again, so retention of body heat and heat loss, so that they can maintain that constant body temperature. So as a result, we're not dependent on absorbing heat from our surroundings like other animals, and we can be active during the day and the night, whatever the external temperature happens to be. So most other animals, with the exception of birds, rely on external sources of heat. Like many cold-blooded amphibians and reptiles will have to lay out, sort of soaking in thermal energy in order to get to their correct internal temperature. The heat that mammals generate is released during respiration, and much of it is produced by liver cells that have a huge requirement for energy. The heat they produce is absorbed by the blood flowing through the liver, and it's distributed around the rest of the body. So again, much of the heat that mammals use, most of the heat that mammals generate is released during respiration, specifically by liver cells, which have this huge requirement for energy. So the heat they produce gets absorbed by the blood flowing through the liver, and then the blood carries it around to the rest of the body. So the hypothalamus in the brain is the central control for body temperature, and it is the body's thermostat, essentially. This region of the brain receives a constant input of sensory information about the temperature of the blood and about the temperature of the surroundings. So the hypothalamus has thermoreceptor cells that continuously monitor this temperature of the blood that's flowing through it. And so as that blood comes from the liver, it circulates through the body, it goes up into the brain. These thermoreceptor cells in the hypothalamus are measuring the temperature of that blood. And the temperature it monitors is the core temperature, the temperature inside the body that remains very close to the set point, which is about 98 degrees Fahrenheit. So this temperature fluctuates a little, but is kept within very narrow limits. And it's done and monitored through the hypothalamus. So the hypothalamus will receive information about temperature from other sources besides these thermoreceptor cells. The skin also contains receptors that monitor change in temperature. And the skin temperature is the first to change if there's a change in the external temperature, right? So if it's cold outside, our skin is the first to feel it. Same if it's hot outside, it's our skin first. So these skin receptors will give this early warning about a possible change in core temperature, which is super, super cool. And if the core temperature does decrease, or the temperature receptors in the skin detect a decrease in the temperature of the surroundings, the hypothalamus will send an impulse that activates the following physiological responses. So vasoconstriction, right? So this is where the muscles in the walls of atrials that supply blood to capillaries near the skin surface will contract. And this narrows the lumens of the atrials and reduces the supply of blood to the capillaries so that less heat is lost from the blood. Okay, and again, these are all things you learned in STEM, so this should be review for you. Okay, the second is shivering. Shivering is the involuntary contraction of skeletal muscles, which generates heat, which is absorbed by the blood and carried around the rest of the body. Also, raising body hairs. Okay, muscles at the base of the hairs in the skin will contract, and it increases the depth of fur or hair, so trapping air close to the skin. Air is a poor conductor of heat, and therefore it's a good insulator. So this is not much use in humans, but it is highly effective for other more hairy mammals. Also decreasing the production of sweat. So this will reduce the loss of heat by evaporation from the skin surface. And increasing the secretion of adrenaline. Okay, so this hormone from the adrenal gland increases the rate of heat production in the liver. The hypothalamus also stimulates higher centers in the brain to bring about some behavioral responses. So some animals will respond by curling up. And it's kind of this like instinctual thing that you do, right? So that's coming from your brain. We respond by finding a source of warmth, putting on warm clothing, curling up. And when an increase in environmental temperature is detected by skin receptors, right, instead of a decrease, so when it gets hot, the hypothalamus will increase the loss of heat from the body and reduced heat production. So it does it in sort of these opposite ways. It does it through vasodilation, right? So instead of vasoconstriction, where you're closing your atrials, you're dilating them. So your muscles in your atrials will relax, which allows more blood to flow through the capillaries, so that more heat is lost to the surroundings. Also lowering body hairs, so muscles attached to the hairs will lie flat, reducing the depth of the fur and the layer of insulation. Increasing sweat production, so sweat glands increase the production of sweat, which will evaporate on the surface of the skin, so removing that heat energy from the body. And the behavioral responses of animals to heat include resting, lying down with our limbs spread out, and of course this increases the body surface that's exposed to the air. We might wear more loose-fitting clothing, turn on fans or air conditioning. We start drinking cold drinks. So when the environmental temperature decreases gradually, as it does with the approach of winter in temperate climates, the hypothalamus releases a hormone which activates the anterior pituitary gland to release thyroid-stimulating hormone, or TSH. Now, TSH stimulates the thyroid gland to secrete the hormone thyroxine into the blood, which increases metabolic rate, which increases heat production, especially in the liver. So when temperatures start to increase again, like towards spring, the hypothalamus responds by reducing the release of TSH by the anterior pituitary gland, so that less thyroxine is released from the thyroid gland. So it's so interesting. So those are things that, immediate reactions that our body will take to keep our internal body temperature correct. And then also these gradual things, like as we're noticing the weather gradually changing. And there are two other examples of the role of negative feedback in homeostasis that we'll talk about later, and that's osmoregulation and blood glucose control. Sometimes control mechanisms don't respond in the way that we've talked about. If a person breathes air that has a very high carbon dioxide content, this can produce a high concentration of carbon dioxide in the blood. This is sensed by carbon dioxide receptors, which cause the breathing rate to increase, right, to get rid of that carbon dioxide. So then the person is breathing even faster, taking in even more carbon dioxide, which stimulates the receptors even more. So then the person breathes faster and faster. And this is an example of a positive feedback. You can see that positive feedback cannot play any role in keeping conditions in the body constant. However, the method is involved in several biological processes, including the transmission of nerve impulses. So many of the metabolic reactions occurring within the body produce unwanted substances, and some of these are toxic. The removal of them is known as excretion. So many excretory products are formed in humans, but two are made in much greater quantities than others. These are carbon dioxide and urea. So carbon dioxide is produced continuously by cells that are respiring aerobically. The waste carbon dioxide is transported from the respiring cells to the lungs and the bloodstream, right? We know this. Gas exchange occurs, and carbon dioxide diffuses from the blood into the alveoli. It's then excreted into the air we breathe out. Urea is produced in the liver. It is produced from excess amino acids, and it's transported from the liver to the kidneys in solution in our blood plasma. So the kidneys will remove urea from the blood and excrete it, of course dissolved in water, as urine. So we're going to look really closely at the production and excretion of urea. If more protein is eaten than is needed, the excess cannot be stored in our body. And it would be wasteful, however, simply to just get rid of it because the amino acids provide us a lot of useful energy. So to make use of it, the liver removes the amino groups in a process known as deamination. This figure shows, well, figure A, shows how deamination takes place. So in the liver cells, the amino group of an amino acid is removed, together with an extra hydrogen atom. And these combine to produce ammonia. The keto acid that remains may enter the Krebs cycle and be respired, or it can get converted to glucose or converted to glycogen for fat storage. We know ammonia is very soluble, but it can be highly toxic. So in many aquatic animals like fish that live in freshwater, ammonia diffuses from the blood and dissolves in the water around them. However, in terrestrial animals like us, ammonia would rapidly build up in our blood and cause immense damage. And damage is prevented by converting ammonia immediately to urea, which is less soluble and less toxic. So there are several reactions known as the urea cycle that are involved in combining ammonia and carbon dioxide to form urea. And these are simplified in B. Down here. So an adult human produces around 25 to 30 grams of urea per day. And it's the main nitrogenous excretory product of humans. We also produce small quantities of other nitrogenous excretory products, mainly creatine and uric acid. A substance called creatine is made in the liver from certain amino acids, but much of the creatine is used in muscles in the form of creatine phosphate, where it acts like an energy store. But most is converted to creatine and excreted. So uric acid is made from the breakdown of purines from nucleotides, not from amino acids. I'm going to say that again. Uric acid is made from the breakdown of purines from nucleotides, not from amino acids. So the urea diffuses from the liver cells into our blood plasma. All of the urea made each day must be excreted, or its concentration in the blood would build up and it would become poisonous. So as the blood passes through the kidneys, the urea gets filtered out and excreted. And to understand how this happens, we have to look at the structure of a kidney. Okay, so this figure here shows the position of the kidneys in the body, and together with their associated structures. So each kidney receives blood from a renal artery and then returns blood via a renal vein. There's a narrow tube called a ureter, which carries urine from the kidney to the bladder. And then from the bladder, a single tube, the urethra, carries urine to the outside of the body. So a longitudinal section through the kidney here shows that it has three main areas. The whole kidney is covered by a fairly tough capsule, beneath which lies the cortex. The central area is made of the medulla. Where the ureter joins, there is an area called the pelvis. So a section through a kidney, seen through a microscope, shows it to be made up of thousands of tiny tubes called nephrons and many, many, many blood vessels. So this shows the position of a single nephron, Figure A shows the position of a single nephron, and B shows its structure. So one end of the tube forms a cup-shaped structure called a Bowman's capsule, which surrounds a tight network of capillaries called a glomerulus. The glomeruli and capsules of all the nephrons are in the cortex of the kidney. Okay, so that's important to understand where their positions are. So here's the Bowman's capsule. From the capsule, the tube runs towards the center of the kidney, first forming this twisted region that's called the proximal convoluted tubule, and then a really long hairpin loop in the medulla called the loop of Henle. And we're going to go into excruciating detail into those. The tubule then runs back upwards into the cortex, where it forms another twisted region called the distal convoluted tubule, before finally joining a collecting duct that leads down through the medulla and into the pelvis of the kidney. Blood vessels are closely associated with the nephrons. Each is supplied with blood by a branch of the renal artery called an afferent arteriole. The capillaries rejoin to form an efferent arteriole. The efferent arteriole leads off to form a network of capillaries running closely alongside the rest of the nephron. And blood from these capillaries flows into a branch of the renal vein. Okay, so the kidney makes urine in a two-stage process. The first stage is ultrafiltration, involving filtering small molecules, including urea, out of the blood and into the Bowman's capsule. From the Bowman's capsule, the molecules flow along the nephron towards the ureter. And the second stage, which is called selective reabsorption, involves taking back any useful molecules from the fluid in the nephron as it flows along. So it's kind of like a check where it's making sure that everything that we are getting rid of into the urea is actually something we want to get rid of and not some nutrient we want to keep. So this figure shows a section through part of a glomerulus and a Bowman's capsule. The blood in the glomerular I struggle with that word, capillary, is separated from the lumen of the Bowman's capsule by 2 cell layers and a basement membrane. The first cell layer is the lining, or the endothelium. Like the endothelium of most other capillaries, this has gaps in it. But there are far more gaps than in other capillaries. Each endothelial cell has thousands of tiny holes in it. So then after that comes the basement membrane, which is made up of a network of collagen and glycoproteins. The second cell layer is formed from epithelial cells, which make up the inner lining of the Bowman's capsule. So these cells have many tiny finger-like projections with gaps in between them, and they're called podocytes. And you can see here is like parts of the podocyte cells. A false color scanning electron micrograph of podocytes shows the blue-green cells with their extensions wrapped around the blood capillary, which is purple. The holes in the capillary endothelium and the gaps between the podocytes are really quite large, much larger than in other cells and other capillaries, and it makes it easier for substances that are dissolved in the blood plasma to get through. However, the basement membrane will stop very large protein molecules from getting through. So any protein molecule with a relative molecular mass of around 69,000 or more won't be able to pass through the basement membrane. And so it cannot escape from the capillary. This basement membrane therefore acts as a filter. Blood cells, both red and white, are also too large to pass, and so remain in the blood. And so this table shows the relative concentrations of substances in the blood and in the glomerular filtrate. You can see that glomerular filtrate is identical to blood plasma, except that there are almost no plasma proteins in it. So the rate at which the fluid filters from the blood in the glomerular capillaries into the Bowman's capsule is called the glomerular filtration rate. In a human, for all the glomeruli in both kidneys, the rate is about 125 cubic centimeters per minute. So what makes the fluid filter so quickly? This is determined by differences in water potential, and it's differences between the plasma in the capillaries and the filtrate in the Bowman's capsule. So you remember that water moves from a region of high water potential to a region of lower water potential down the water potential gradient. Water potential is lowered by the presence of solutes, right? And it's raised by high pressures. So inside the capillaries in the glomerulus, the blood pressure is relatively high, because the diameter of the afferent arteriole is wider than that of the efferent arteriole, causing a head of pressure inside the glomerulus. This tends to raise the water potential of the blood plasma above the water potential of the contents in the Bowman's capsule. However, the concentration of solutes in the blood plasma in the capillaries is higher than the concentration of solutes in the filtrate. So this is because while most of the contents of the blood plasma filter through the basement membrane, right, the plasma protein molecules are too big to get through, and so they stay in the blood. So this difference in solute concentration tends to make the water potential in the blood capillaries lower than that of the filtrate in the Bowman's capsule. Overall, though, the effect of differences in pressure outweighs the effect of differences in solute concentration. Overall, the water potential of blood plasma in the glomerulus is higher than the water potential of the filtrate. So water continues to move down the water potential gradient from the blood into the capsule. Many of the substances in the glomerular filtrate need to be kept in the body, so they are reabsorbed into the blood as the fluid passes along the nephron. But only certain substances are reabsorbed, and the process is called selective reabsorption. Most of the reabsorption takes place in the proximal convoluted tubule. That's the lining in this part of the nephron, and it's made of a single layer of cuboidal epithelial cells. And these cells are adapted for their function of reabsorption by having microvilli to increase the surface area, tight junctions that hold adjacent cells together so that fluid can't pass between them, many mitochondria to provide energy, which you need for the sodium-potassium pump, co-transporter proteins in the membrane facing the lumen. So blood capillaries here are very close to the outer surface of the tubule. The blood in these capillaries has come directly from the glomerulus, so it has much less plasma in it than it did before. The basal membranes of the cells lining the proximal convoluted tubule are those nearest to the blood capillaries. So sodium-potassium pumps in these membranes move sodium ions out of the cell. The sodium ions are carried away in the blood, so lowering the concentration of those sodium ions inside the cell so that they passively diffuse into it down their concentration gradient from the fluid in the lumen of the tubule. Sodium ions don't diffuse freely through the membrane, though. They can only enter through special co-transporter proteins in the membrane. And there are several different kinds of these proteins, each of which transports something different, such as glucose or amino acids, but at the same time as the sodium ion. So remember, from AS biology, that's why they're called co-transporter proteins, because their specific purpose is for glucose or something else, but those sodium ions can get through as well. So this passive movement of sodium ions into the cell down their concentration gradient provides that energy to move the glucose molecules, even against a concentration gradient. And the movement of the glucose and of the other solutes is an example of indirect or secondary active transport, since the energy is used in the pumping of sodium ions, not in the moving. So once inside the cell, glucose diffuses down its concentration gradient through a transport protein in the basal membrane into the blood. All of the glucose in the glomerular filtrate is transported out of the proximal convoluted tubule and into the blood. Normally, no glucose is left in the filtrate, so no glucose is present in urine. Similarly, amino acids, vitamins, and other sodium and chloride ions are reabsorbed in the proximal convoluted tubule. And the removal of these solutes from the filtrate greatly increases the water potential, right? Because you're removing the solutes. So the movement of the solutes into the cell and into the blood decreases water potential, and so then that gradient exists between the filtrate and the blood. Water moves down the gradient through the cells and into the blood. So the water and reabsorbed solutes are carried away back into the circulation. And surprisingly, quite a lot of urea is reabsorbed too, actually. Urea is a small molecule and it can pass easily through cell membranes. Its concentration in the filtrate is considerably higher than that in the capillaries, so it diffuses passively through the cells of the convoluted tubule and right back into the blood. Actually, about half of the urea in the filtrate is reabsorbed in this way. The other two nitrogenous excretory products, uric acid and creatine, are not reabsorbed. Creatine is actively secreted by the cells of the proximal convoluted tubule into its lumen. So the reabsorption of so much water and solutes from the filtrate in the proximal convoluted tubule greatly reduces the volume of liquid remaining. So in an adult human, about 125 cubic centimeters of fluid enter the proximal tubules every minute, but only about 64% of it passes on to the next region of the nephron into the loop of Henle. So again, let's really look at this model. So here you have your sodium-potassium pumps in the basal membrane of proximal convoluted tubule cells, and they use ATP made by the mitochondria. So these pumps decrease the concentration of sodium ions in the cytoplasm. The basal membrane is folded to give a large surface area for many of these carrier proteins. Very close by, the blood plasma rapidly removes absorbed sodium and chloride, glucose, and amino acids. So this helps further uptake from the lumen of the tubule. And then microvilli will increasing that surface area, helping the uptake of solutes. And sodium ions move passively into the cell down its concentration gradient. It moves in using these protein co-transporter molecules in the membrane, which bring in glucose and amino acids as well as those sodium ions. About 1/3 of our nephrons have these long loops of Henle, and these dip down into the medulla. The function of these long loops is to create a very high concentration of sodium and chloride ions in the tissue fluid in the medulla. So as you can see, this enables a lot of water to be absorbed from the fluid in the collecting duct as it follows through the medulla. This allows the production of very concentrated urine, which means that water is being conserved in the body rather than lost, which helps us to prevent dehydration. So you can see here in this model of the loop of Henle, the hairpin loop runs deep into the medulla of the kidney before turning back towards the cortex again. The first part of the loop is the descending limb, and the second part is the ascending limb. And these differ in their permeabilities to water, which is really interesting. The descending limb is permeable to water, whereas the ascending limb is not. And to explain how the loop of Henle works, it's best to start by describing what happens in the ascending limb. So the cells that line this region of the loop actively transport sodium and chlorine atoms out of the fluid in the loop into the tissue fluid. And this decreases the water potential in the tissue fluid and increases the water potential of the fluid outside. The cells lining the descending limb are permeable to water and also to sodium chloride ions. And as the fluid flows down the loop, water from the filtrate moves down a water potential gradient into the tissue fluid by osmosis. And at the same time, those sodium and chloride ions will diffuse into the loop down their concentration gradient. So by the time the fluid has reached the very bottom of the hairpin, it contains much less water and many more sodium and chloride ions than it did when it entered. So the fluid becomes more concentrated towards the bottom. And the longer the loop, the more concentrated the fluid can become. The concentration in human kidneys can be as much as 4 times the concentration of blood plasma. This concentrated fluid flows up the ascending limb, and as the fluid inside the loop is so concentrated, it is relatively easy for sodium and chloride ions to leave it and pass into the tissue fluid, even though the concentration in the tissue fluid is also pretty high. So, especially high concentrations of sodium and chloride ions can be built up in the tissue fluid between the two limbs near the top of the loop. As the fluid continues up the ascending limb, losing sodium and chloride ions the whole way, it becomes gradually less concentrated. However, it is still relatively easy for sodium ions and chloride ions to be actively removed because these higher parts of the ascending loops are next to concentrated regions of tissue fluid. So all the way up, the concentration of sodium and chloride ions inside the tubule is never very different from the concentration in the tissue fluid. So it is never too difficult to pump sodium and chloride ions out of the tubule into the tissue fluid. So having the two limbs of the loop running side by side like this, with the fluid flowing down in one and up in the other, enables the maximum concentration of solutes to be built up, both inside and outside the tube at the bottom of that loop. And this mechanism is called a countercurrent multiplier. But it's not done yet. You've seen that the fluid flowing up the ascending limb of the loop of Henle loses sodium and chloride ions, so becoming more dilute and having a higher water potential. And so the cells of the ascending limb of the loop of Henle, the cells lining the collecting ducts, are permeable to that urea, so which it will diffuse into the tissue fluid. And as a result of that, urea is also concentrated in the tissue fluid in the medulla. So here you can see that the fluid continues round through the distal convoluted tubule into the collecting duct and then runs down into the medulla again. So really it passes once again through the regions where the solute concentration of the tissue fluid is very high and the water potential is very low. Water therefore can move out of the collecting duct by osmosis until the water potential of urine is the same as the water potential of the tissue fluid in the medulla, which may be greater than the water potential of the blood. So the degree to which this happens is controlled by ADH, or antidiuretic hormone. So the ability of some mammals, like some rodents, to produce a very concentrated urine is related to the relative thickness of the medulla in their kidneys. Desert rodents like gerbils and kangaroo rats can produce a urine that is about 20 times the concentration of their blood plasma. And it's only possible because the medulla is really large in ratio, and the cells that line the ascending limb of their loops have really deep folds with a lot of sodium and potassium pumps, and cytoplasm filled with tons of mitochondria, each that have many cristae that allow the production of a lot of energy for the pumping of sodium ions into the tissue fluid. So the first part of the distal convoluted tubule functions in the same way as the ascending limb of the loop of the Henle. The second part functions in the same way as the collecting duct. So the functions of this part of the distal convoluted tubule and the collecting duct will be described together. In the distal convoluted tubule and collecting duct, sodium ions are actively pumped from the fluid in the tubule into the tissue. From there, they'll pass into the blood. Potassium ions, however, are actively transported into the tubule, so the rate at which they are moved in and out of the fluid in the nephron can be varied and helps to regulate the concentration of the ions in the blood. Osmoregulation is the control of the water potential of body fluids. So this regulation is an important part of homeostasis, and it involves the hypothalamus, the posterior pituitary gland, and the kidneys. The water potential of the blood is constantly monitored by these specialized sensory neurons in the hypothalamus called osmoreceptors. And when these cells detect a decrease in water potential of the blood, the nerve impulses are sent along neurons to where they terminate in the posterior pituitary gland. Now, these impulses will stimulate the release of antidiuretic hormone, which is a peptide hormone made of 9 amino acids. And molecules of this ADH will enter the blood in the capillaries and are carried all around the body. And the effect is to reduce the loss of water in the urine by making the kidney reabsorb as much water as it can. So the word diuresis means the production of dilute urine. Antidiuretic hormone gets its name because it stops dilute urine from being produced by stimulating the reabsorption of that water. So water is reabsorbed by osmosis from the fluid in the nephron as the fluid passes through the collecting ducts. The cells of these collecting ducts are target cells for ADH. As you can see in the image, this hormone acts on the cell surface membranes of the collecting duct cells, making them more permeable to water than usual. And remember, at any time, you should be pausing this to really study these models, and most of these models come from your book, so you can study them there as well. So this change in permeability is brought about by increasing the number of the water permeable channels, known as aquaporins, in the cell surface membrane of the collecting ducts. So ADH molecules bind to receptor proteins on the cell surface membranes, which in turn activate enzymes inside the cells. And the cells contain ready-made vesicles that have many aquaporins in their membranes. And so once the enzymes in each cell are activated by the arrival of ADH, these vesicles move towards the cell surface membrane and fuse with it, so increasing the permeability of the membrane to water. ADH binds to receptors in the cell surface membranes of the cells lining the ducts. This activates a series of enzyme-controlled reactions. The phosphorylase causes vesicles surrounded by those membranes to move to the cell surface membrane. They bond with it fuses with the cell surface membrane, and then thus becomes more permeable. So as the fluid flows down through those collecting ducts, water molecules move through the aquaporins out of the tubule and, of course, back into the tissue fluid. This happens because the tissue fluid in the medulla has a very low water potential, and the fluid in the collecting ducts, of course, has a high water potential. So the fluid in the collecting duct loses water and becomes more concentrated. And the secretion of ADH has caused the increased absorption of water into the blood. So the volume of urine, which flows from the kidneys into the bladder, will be smaller, and the urine will be more concentrated. What happens when you have more than enough water? So that was when we needed more water, right? So ADH is increasing the amount of water that we're retaining from our urine to keep us from dehydrating. But what happens when we have enough water? When there is an increase in water potential of the blood, those same osmoreceptors in the hypothalamus are no longer stimulated, and the neurons in the posterior pituitary gland stop secreting ADH. And this affects the cells that line the collecting ducts, so the aquaporins are removed out of the cell surface membrane back into the cytoplasm as part of the vesicles. And this makes the collecting duct less permeable. So the fluid flows down the collecting duct without losing water. So then a dilute urine collects in the pelvis and flows down the ureter to the bladder. And under those conditions, we tend to produce larger volumes of dilute urine, losing much of the water we drink in order to keep our water potential constant. So the collecting duct cells do not respond immediately to the reduction in ADH secretion by the posterior pituitary gland. And it's because it takes time for the ADH already in the blood to be broken down. So approximately half of it will be destroyed every 15 to 20 minutes. However, once ADH stops arriving at the collecting ducts, it takes only 10 to 15 minutes for aquaporins to be removed from that cell surface membrane. Carbohydrate is transported through the human bloodstream in the form of glucose in solution in the blood plasma. And glucose is converted into the polysaccharide glycogen, a large insoluble molecule made up of many glucose units linked together by 1,4 glycosidic bonds with 1,6 branching points, which you guys know all about from AS Bio. Glycogen is a short-term energy store that is found in liver and muscle cells, and it's easily converted back to glucose. So in a healthy human, each 100 cubic centimeters of blood contains about 80 to 120 milligrams of glucose. If the concentration decreases below that, cells may not have enough for respiration, and they're unable to carry out their normal activities. This is especially important for cells that can respire only glucose, like brain cells. Very high concentration of glucose in the blood can also cause problems. Again, it upsets the normal behavior of cells. So the homeostatic control of blood glucose concentration is carried out by two hormones secreted by endocrine tissue in the pancreas. This tissue consists of groups of cells known as the islets of Langerhans, which are scattered throughout the pancreas. And the word islet means small island, as you might find in like a river. The islets contain two types of cells: alpha cells, which secrete glucagon, and beta cells, which secrete insulin. So the alpha and beta cells act as receptors and the central control for the homeostatic mechanism. The hormones glucagon and insulin coordinate the actions of the effectors. So here you see how the blood glucose concentration fluctuates within normal limits around that set point, right? So here's the set point, and then here's our concentration, and it's ebbing and flowing. After a meal, where you're eating a lot of carbohydrates, glucose from that digested food is absorbed from the small intestine and then passes into the blood. As this blood flows through the pancreas, the alpha and beta cells will detect the increase in glucose concentration. So the alpha cells will respond by stopping the secretion of glucagon, whereas the beta cells respond by secreting insulin into the blood plasma. Insulin is carried to all parts of the body through the blood. And insulin is a signaling molecule. So as it is a protein, it can't pass through cell membranes to stimulate mechanisms directly, but it can bind to a receptor in the cell surface membrane, and it can affect the cell indirectly through the mediation of intracellular messengers. So there are insulin receptors on many cells, such as those in the liver, muscle, and adipose tissue. Insulin stimulates cells with these receptors to increase the rate at which they can absorb glucose from the blood, convert it into glycogen, and then use it for respiration. And this results in a decrease in the concentration of glucose in the blood. It can only enter cells through transporter proteins known as GLUT. Okay, there are several different types of GLUT proteins. Muscle cells have the type called GLUT4 or GLUT4. Normally, the GLUT proteins are kept in the cytoplasm in the same way as the aquaporins are in the collecting ducts. So when insulin molecules bind to receptors on those muscle cells, The vesicles with GLUT4 proteins are moved to the cell surface membrane, they fuse with it, and this will facilitate the movement of glucose into the cell. Brain cells have GLUT1 proteins, and liver cells have GLUT2, which are always in the cell surface membrane, and their distribution is not altered by insulin. Insulin also stimulates the activation of the enzyme glucokinase, which phosphorylates glucose. And this will trap glucose inside cells because phosphorylated glucose cannot pass through the transporters in the cell surface membrane. Insulin will also stimulate the activation of two other enzymes, which together add glucose molecules to glycogen. And this increases the size of the glycogen granules inside the cell. Oh, shoot. Insulin also stimulates the activation of the enzyme glucokinase, which phosphorylates glucose. This traps glucose inside cells because phosphorylated glucose can't pass through the transporters in the cell surface membrane. Insulin will also stimulate the activation of two other enzymes, which together add glucose molecules to glycogen. And this increases the overall size of those glycogen granules inside the cell. A decrease in blood glucose concentration is detected by the alpha and beta cells in the pancreas. So the alpha cells respond by secreting glucagon, while the beta cells respond by stopping the secretion of insulin. The decrease in the concentration of insulin in the blood reduces the rate of uptake and the use of glucose by liver and muscle cells. Uptake will still continue, but at a lower rate. So glucogen binds to different receptor molecules in the cell surface membranes of liver cells. The method of cell signaling is the same as described earlier in, chapter 4 or 5 when we talk about cell signaling and cell surface membranes. The binding of glucagon to a receptor activates that G protein that in turn activates an enzyme within the membrane that will then catalyze the conversion of ATP to cyclic AMP, which is that second messenger. So the cyclic AMP binds to kinase enzymes within the cytoplasm that activate other enzymes. Kinase enzymes activate enzymes by adding phosphate groups to them in a process known as phosphorylation. And this enzyme cascade amplifies the original signal from glucagon. Glycogen phosphorylase is at the end of that enzyme cascade. When it's activated, it catalyzes the breakdown of glycogen to glucose. And it does this by removing glucose units from the numerous ends of glycogen. So this increases the concentration of glucose inside the cell so that it can diffuse out through GLUT2 transporter proteins back into the blood. And glucose is also made from amino acids and lipids in a process known as glucogenesis, which literally means the formation of glucose. And as a result of glucagon secretion, the liver will release extra glucose to increase the concentration in the blood. Muscle cells don't have receptors for glucagon and so do not respond to it. Glucagon and insulin work together as part of the negative feedback system, in which any deviation of the blood glucose concentration from the set point will stimulate actions by those effectors to bring it back to normal. Blood glucose concentrations never are constant, even in the healthiest person, and one reason is the inevitable time delay between the change in blood glucose concentration and the onset of the actions to correct it. Time delays in control systems result in that oscillation, where things do not stay at an absolute constant, but sometimes go slightly above and sometimes slightly below the required level. So the hormone adrenaline also increases the concentration of blood glucose, and it does this by binding to different receptors on the surface of liver cells that activate the same enzyme cascade and lead to the same end result. And the breakdown of glycogen by glycogen phosphorylase. Adrenaline also stimulates the breakdown of glycogen stores in muscle during exercise. So the glucose produced remains in the muscle cells where it's needed for respiration. So sugar diabetes, or diabetes mellitus, is one of the most common metabolic diseases in humans. In 2013, the International Diabetes Federation estimated that 382 million people, or 8.3% of the world's population, had it, and that number is increasing. So although the percentages are higher among some ethnic groups, it is a disease that is increasing steeply everywhere. There are two forms of sugar diabetes, an insulin-dependent one, known as type 1 diabetes. The pancreas seems to be incapable of secreting sufficient insulin. And it's thought that this might be due to a deficiency in the gene that codes for the production of insulin, or because of an attack on the cells by the person's own immune system, and it's an attack on the beta cells. So type 1 diabetes is sometimes called juvenile onset diabetes because it usually begins pretty early. The second form is called non-insulin dependent diabetes, or type 2 diabetes. In this form of diabetes, the pancreas doesn't secrete insulin, but the liver and muscle cells don't respond properly to it. I'm sorry, let me repeat that. In this form of diabetes, the pancreas does secrete insulin, but the liver and muscle cells don't respond properly. Type 2 diabetes begins relatively late in life and is often associated with poor diet and obesity. So the symptoms of both types of diabetes mellitus are the same. After a carbohydrate meal, glucose is absorbed into the blood and the concentration increases and stays high. Normally, there is no glucose in urine, but if the glucose concentration in the blood becomes very high, the kidney cannot reabsorb all the glucose, so that some passes out in the urine. Extra water and salts accompany the glucose, and the person consequently feels extremely hungry and thirsty. In a diabetic person, uptake of glucose into cells is slow, even when there's plenty of glucose available. And thus the cells lack glucose and metabolize fats and proteins as alternative energy sources. And this can lead to a buildup of substance in the blood called ketoacids or ketones. And these are produced when the body switches to metabolizing fat and they decrease the blood pH. So the combination of dehydration, salt loss, and low blood pH causes coma in extreme situations. So between meals, the blood glucose concentration of a person with untreated diabetes decreases steeply. And this is because there's no glycogen to metabolize, as it was not stored when there was plenty of glucose. So once again, coma may result, this time because of a lack of glucose for respiration. People with type 1 diabetes receive regular injections of insulin, which they have to learn to do themselves. They must also take blood samples to check that the insulin is actually working. Some people have mini pumps, which deliver the exact volumes of insulin they need, and a carefully controlled diet can also help to maintain a near constant concentration of glucose in the blood. People with type 2 diabetes rarely need to have insulin injections. Instead, they can use diet and regular and frequent exercise to keep their blood glucose within normal limits. Diabetics now receive insulin made by genetically engineered cells. So it's much easier to collect a urine sample from someone than a blood sample. Simple tests on urine can give early indications of health problems. The presence of glucose and ketones in urine can indicate that a person may have diabetes. And if blood glucose concentration increases above a certain value, known as the renal threshold, not all of the glucose is then reabsorbed from the filtrate in the proximal convoluted tubule of the kidney, and then some will be present in the urine. So the presence of this protein in the urine indicates that there is something wrong with the kidneys. And most protein molecules being too large to be filtered, but some are, and these are reabsorbed by endocytosis in the proximal convoluted tubule, broken down, and then those amino acids get absorbed into the blood. So it's not unusual for some protein to be present in the urine for short periods of time, such as during a fever, after vigorous exercises, and also during pregnancy. But a large quantity over long term indicates that the urine can have a disease affecting the glomeruli, or there is a kidney infection. Protein in the urine is also associated with high blood pressure, which is a risk factor in heart disease. So dipsticks or test strips can be used to test urine for a range of different factors, including pH, glucose, ketones, and proteins. Dipsticks for detecting glucose contain the enzymes glucose oxidase and peroxidase. These two enzymes are immobilized on a small pad at one end of a stick. The pad is immersed in the urine, and if it contains glucose, glucose oxidase catalyzes a chemical reaction, and the hydrogen peroxide is then produced. So this peroxidase catalyzes a reaction between hydrogen peroxide and a colorless chemical in the pad. and it will form a brown compound. So the resulting color of the pad is matched against a color chart. The chart shows the colors that indicate different concentrations of glucose. The more is present, the darker the color. So one problem, though, is that they don't indicate current blood glucose concentration, only whether the concentration was higher than the renal threshold in the period of time when the urine was collecting in the bladder. So a biosensor allows people with diabetes to check their blood immediately. And like the dipstick, they use a pad impregnated with glucose oxidase, but a small sample of blood is placed into the machine. And in the same way, it catalyzes a reaction. The current is detected, and at the same time, a tiny electric current is generated. That current is detected by an electrode, amplified, and read by the meter, which produces a reading for blood glucose. The more glucose that is present, the greater the current, and the greater the reading from the biosensor. So, just to close here, it's important to remember that plants also have to maintain constant internal environment. For example, mesophyll cells and leaves require a constant supply of carbon dioxide if they're going to use the best of the light energy for photosynthesis. So we've seen that low concentrations of carbon dioxide limit the rate of photosynthesis. Stomata control the entry of carbon dioxide in leaves. Strictly speaking, a stoma is the hole between the guard cells, but this term is usually used to refer to the two guard cells and the hole between them. But stomata look simple, but the guard cells are highly specialized and they respond to a wide range of environmental stimuli. And so therefore they can be used as a control for the internal atmosphere of the leaf. Stomata show daily rhythms of opening and closing. So even when kept in constant light or constant dark, their rhythms will persist. They open during the day, maintaining the inward diffusion of carbon dioxide and the outward diffusion of oxygen. And it also allows the outward diffusion of water vapor. And we know that it's a part of transpiration. The closure of the stomata at night, when photosynthesis isn't occurring, can reduce the rate of transpiration and therefore conserve water. Stomata respond to changes in environmental conditions. They open in response to increasing light intensity, low carbon dioxide concentrations as well. So when those stomata are open, leaves gain the carbon dioxide for photosynthesis, but they also tend to lose water. And then also stomata will close in response to darkness, high carbon dioxide concentrations, low humidity, high temperature, and water stress. So each stomatal pore is surrounded by two guard cells, and they'll open when they gain water to become turgid, and then they close when they lose water to become flaccid. Guard cells gain and lose water by osmosis, and a decrease in water potential is needed before water can enter the cells by osmosis. This is brought about by the activities of transporter proteins in their cell surface membranes. These ATP-powered proton pumps in the membrane actively support hydrogen ions, and the decrease in the hydrogen ion concentration in the cells causes these channel proteins in the cell surface membranes to open, and so that those potassium ions can move through the cell. And we know that. We've learned a bit about that last year. They do this because the removal of hydrogen ions has left the inside of the cell negatively charged compared with the outside, and as those potassium ions have a positive charge, they're drawn down the electrical gradient towards the negatively charged region. They also diffuse into the cells down a concentration gradient. Such a combined gradient is an electrochemical gradient. The extra potassium ions inside the guard cells lower the solute potential and therefore the water potential. Now there is a water potential gradient between the outside and the inside of the cell, so water moves in by osmosis through aquaporins in the membrane. This increases the turgor of the guard cells and the stoma opens. So these guard cells have unevenly thickened cell walls. The cell wall adjacent to the pore is super thick, whereas the wall furthest from the pore is very thin. Bundles of cellulose microfibrils are arranged as hoops around the cell so that as the cell becomes turgid, these hoops ensure that the cell mostly increases in length and not diameter. So since the ends of the two guard cells are joined and the thin outer walls bend more readily, the thick inner walls become curved. So this opens the pore between the two cells. Stomata close when the hydrogen ion pump proteins stop and potassium ions leave the guard cells and enter neighboring ones. So now this creates that reverse water potential gradient, So water is leaving the guard cells and they're becoming more flaccid, and then the stoma closes. So closing of the stomata has significant effects on the plant, right? It reduces the uptake of carbon dioxide, and it reduces the rate of transpiration. So as transpiration is needed or used for cooling the plant and also for maintaining the transpiration stream, stomatal closure only occurs when reducing the loss of water vapor. Conserving water is more important. In conditions of water stress, the hormone abscisic acid is produced in plants to stimulate stomatal closure. The ABA, which is the abscisic acid, and stomatal closure have been found in a very wide variety of plants, including ferns and mosses. Okay. One role of ABA is to coordinate the responses to stress, and it is, a stress hormone. So if a plant is subjected to difficult environmental conditions, like high temperatures or reduced water, it responds by secreting that ABA. In a plant in drought conditions, the concentration of ABA in the leaves can rise to 40 times that would normally be present. This high concentration will stimulate those stomata to close too, so that the loss of water vapor is reduced. So if the ABA is applied to a leaf, then the stomata will close within just a few minutes. It's not exactly known how ABA achieves the closure of stomata, but it seems like the guard cells have ABA receptors on their cell surface, and that so then it's possible that when the ABA binds with these, it inhibits the proton pumps. It also stimulates the movement of calcium ions into the cytoplasm. And calcium acts as a second messenger, which activates channel proteins to open, and that allows negatively charged ions to leave the guard cells. So this in turn will stimulate the opening of the channel proteins, which allows the movement of potassium ions out of the cells. And at the same time, calcium ions also stimulate the closure of the channel proteins that allow potassium ions to enter. And the loss of these ions raises the water potential, which passes out by osmosis. And then those guard cells can become flaccid, and the stomata can close again. Okay. So there is a lot of detail in this chapter, and it's going to take, really studying and memorizing all of these models and diagrams and all of the details involved. And I will see you in class.